Dear colleagues, ladies and gentlemen, it is my pleasure to be here. And first of all, I would like to thank the organizers inviting me to this very prestigious symposium. I'm particularly grateful to Zsuzsa Helyes and Peter Ferdinand giving me the opportunity to speak here. So in my talk, I would like to focus to the design of new ligands for G-protein coupled receptors. And I would like to introduce you a novel methodology that how exactly we can combine different molecular fragments to achieve the desired pharmacology. But at the very beginning, I would like to convince you that why exactly GPCRs are so interesting. So you know that this is clearly one of the biggest class of drugs actually targeting different GPCRs. So more than 500 FDA-approved drugs already targeting a little bit more than 100 unique GPCRs. And we have many, many other opportunities still open. Just please consider the relatively large number of targets, especially of those GPCRs that are typically orphan. And this type of basic research, just addressing the orphan receptors, would help us to identify new drug targets and to facilitate GPCR-based drug discovery. So our motivation is coming from the new opportunities associated with the GPCR research. I would like to remind you the very important discoveries associated with the multiple signaling pathways in GPCR pharmacology. Also, a large number of different binding sites available at the surface of these proteins. Not only the orthosteric, but many, many allosteric sites are available for pharmacological intervention. And also, we have a limited knowledge on the dimerization and oligomerization of these receptors. However, there are clear indications that these processes are involved heavily in the pharmacological response through different GPCRs. For us medicinal chemists, we are especially motivated with a pretty large number of structures actually available on different GPCRs. And in addition to Rob Lefkowitz, that contributed a lot to GPCR pharmacology, I would like to mention Brian Kobilko. They shared the Chemistry Nobel Prize in 2012. Brian Kobilko contributed to the discovery of novel methodologies that helped a lot to determine different GPCR structures. So just let's have a look at the general structure of the GPCRs. So basically, there are two modules I would like to show you. The upper part, this is the extracellular part of the GPCRs that are typically located in the membrane. This is the ligand binding module that typically shows higher diversity, higher structural diversity, and on the other hand, we typically observed a smaller conformational changes associated with the ligand binding. On the other hand, there is a downstream signaling module here that is located in the intracellular part of these proteins. It shows typically lower diversity between the different GPCRs. However, there are significant conformational changes associated with the activation and inactivation process of different GPCRs. So this is a wonderful machinery, and that's why I call these pieces as the Magnificent Seven. We analyzed a little bit of the structures available for different Class A GPCRs, and I would like to show you a couple of structures, focusing first on the beta-1, beta-2 receptors. These are already mentioned by our invited speakers. And if you have a short look on that you could realize that this is the binding site typically used by the endogenous molecules. And there is a large empty space above the orthosteric binding site that would be available for further modification of the ligand. if you see here the adenosine A2A receptor, then the orthosteric binding site is filled by the ligand. However, it is extended towards the secondary binding site, that is the equivalent of the large empty space observed in the case of the adrenergic receptors. Interestingly, in the case of the rhodopsin receptor, you could not see any empty space above the orthosteric binding site. In the case of the histamine H1 receptor, there is a large empty space here. However, you could see that here is a phosphate ion we can observe, and this is, phosphate ion is coming from the crystallization buffer. However, it clearly shows that this space might be available to extend the original ligand, the antihistamine, here. And this is also the case in the dopamine D3 receptor. Here you could see ethylchloride sitting in the bottom of the pretty large binding site, and here is the empty space that we call the secondary binding site that is available for further ligand development. The problem is that unfortunately this upper binding site is a metastable site. So you simply could not use typically the experimental screening methodology to identify molecules that bind exclusively to the secondary binding site. However, it would be very useful because if you see this big table, and please concentrate only on the colors. So if you see that the blue amino acid positions, these show low conservation, and most of the blue and the green, the medium conserved amino acid residues, these are located in the secondary binding site. And we have much less option to develop selective molecules if we are focusing only to the orthosteric side. So this very simple analysis suggests that if we extend our molecules towards the secondary binding site, that would be a very good strategy to improve not only the potency of the compounds, just generating more interactions with the receptor, but also to improve the specificity, the selectivity of the compounds. So the problem I mentioned, unfortunately, the secondary binding packet, this is a temporary and metastable binding site. So that's why we developed partly experimental and partly computational screening technology to identify molecular fragments that could bind to the secondary and the orthosteric binding site. In the case of the orthosteric site, of course, we can use experimental screening, and small molecular fragments we use typically in this type of screening experiments to identify compounds having a significant binding affinity towards this orthosteric site. And now the trick comes because during the computational part of the protocol, we set up the target protein, the target GPCR, with molecular fragments already bound to the orthosteric binding site, and we try to identify computationally those small fragments that can bind in the presence of the orthosteric part of the molecule. So this is the protocol. So first we screen orthosteric fragments, and then we set a virtual fragment library, and we use the docking methodology to try a different SBP, secondary binding packet fragments, in the presence of the OBP binders. And when we identify these suitable pairs of OBP and SBP binding molecules, the last step was just trying to link these two small fragments to identify the so-called bitopic ligands. Bitopic, it means that the molecule now binds both of the binding sites, the orthosteric and the secondary side. So I would like to show you a couple of examples that how exactly we can control potency and selectivity from the secondary binding side. The first example would be trivially, at least in my case, dopamine D2 and D3 receptors. We worked a lot together with the scientists during the discovery of cariprazine and other compounds. So just let me show you first the high-resolution structure of dopamine D2 receptor complex with risperidone, a very well-known second-generation antipsychotics. And this is actually a bitopic molecule. This one, this part, this piperidine part, this is responsible for mimicking the dopamine, and that's why this part of the molecule is sitting exactly at the same place where dopamine binds. However, just let's see, this molecule reaches also the secondary binding site with this bicyclic component. And that's why the molecule, the risperidone, is a selective D2 antagonist. What is the situation with the D3 receptor? This is the molecule complex of the D3 receptor co-crystallized with ethylchloride. Ethylchloride, this is a very small drug, and this is located exactly in the same place where dopamine does. So in the case of the D3 receptor, we might have typically the same opportunity, just extending the molecules toward the secondary binding site. Okay, so this is a summary of what we did. First, we screened an oral piperazine library experimentally and trying to identify those compounds, oral piperazines, all mimicking the dopamine, the endogenous ligand, to identify the best OBP binders. And then we compiled this set of small fragments, molecular fragments, and we used the computational screening to identify the best pairs of orthosteric and secondary site binders. In this slide, you could see the results of this type of analysis. And here are the oral piperazines and oral piperidines we identify binding best to the dopamine D3 receptor. And in the case of the SBP binders, we found typically the cyclohexylamides that were the best in the presence of these molecules. So now the next step was just trying to connect these two pieces into one single molecule. And you can see here the binding site occupied with the oral piperazine moiety here, and the binding site here that is occupied by the cyclohexyl amide part of the fragment. And you could see some critical interactions. We identified that these interactions are responsible for the selective D3 binding of the molecule. So let me show you that when picking up this OBP binder, the dopamine mimetic, this showed originally the D2 preference. However, if we connect it to this molecule that is located in the SBP1, the secondary binding site, then we observed a balanced compound. However, if we move further and changed the fragment that is responsible for the interactions with the secondary site, you could see that there is a significant improvement in the D3 preference. Okay, pretty much the similar trends we observed with the other OBP binders here. This OBP binder was a balanced compound, and its D3 selectivity was improved by both of the SBP binders when we connected these fragments to the OBP2 binder. And finally, this compound alone showed some preference for the D3 receptor. However, we were able to increase the D3 preference significantly when extending the molecule towards the secondary binding packet. Why it is important? So here is coriprozin bound to the D3 receptor. You could see the two most important interactions that are stabilizing the drug within the binding site. One is the pretty much conserved aspartate residue that is available virtually all of the class A GPCRs. So this is typically not the interaction that would provide you the needed selectivity. However, there is another glutamate over here that is located at the secondary binding site, and this is also responsible for forming a stable hydrogen bond with the coriprozin structure. And this provided some preference in the case of coriprozin towards the D3 receptor. So now we developed a little bit the SBP binding moiety further, just replacing the carboxamide to the sulfonamide. And look at that. What happened, now we were able to create two hydrogen bonds with the same glutamate here, and it improved pretty much the selectivity. And the very fresh results from our laboratory can be seen in the last screenshot here. So we extended the SBP binding part of the molecule a little bit further, and we were able to improve the selectivity of the compounds toward the D3 receptor almost 500-fold. And the big achievement is that, look at that. So basically, the compound shows the same affinity as coriprozin. However, in the functional assay, we observed a much higher potency associated with the modified version of the compound. Okay, probably you would be interested in it. Okay, it works for the dopamine receptor. What happens with the others? Type of receptors? I would like to give you just two more examples. One example is the histamine H1 receptor. This is doxapine, located in the orthosteric binding site, and this is the famous phosphate ion located in the secondary binding site. And the problem is, typically with the first generation antihistamines, that there is a side effect associated with the M1 receptor binding, and this is salivation. So that's why the challenge is that how exactly we can improve the histamine H1 M1 selectivity. So we started from this OBP binding structure that showed some minor preference for the H1 receptor, and look at that. When we use this computational screening and identify these fragments that were connected to the orthosteric binder here, we were able to improve the H1 specificity very significantly. In the case of the SBP2 fragment, more than 100-fold selectivity was observed. This is a challenging case, very challenging case. I would like to show you the 5-HT1B2B receptors showing really high homology, not only in the case of the OBP, but also the SBP binding moieties. So the question was how exactly we can develop a molecule with a 2B preference. So we started from the LSD here, that shows a 10-fold 5-HTB preference. This is an ergotamine, a natural product that was already extended towards the SBP binding packet by nature. However, unfortunately, ergotamine did not show any preference for the 5-HT2B receptor. But instead, if we replaced this moiety of ergotamine with a computationally identified small fragments connected to pretty much the same scaffold, then we were able to observe some significant shift in the 5-HTB preference. If you see the general pharmacology on other receptors as well, it is pretty much interesting that using this computational protocol, especially in the case of the SBP S2 we were able to improve the overall selectivity of the compound towards 5-HT2B receptors. In the next part of my talk, I would like to focus at how exactly we can control now the functional activity and the functional selectivity from the SBP. I think that you are pretty much familiar with the G-protein coupled receptor signalization pathways, especially the G-protein signaling and also the other option that is based on the beta-arrestin recruitment. So what we did, we investigated a set of compounds in both of the G protein and the beta-arrestin assay. And I would like to show you that starting with this small fragment that binds exclusively in the orthosteric site of the dopamine D3 receptor, then we found that this is a weak antagonist. If we extended this molecule towards the secondary binding packet with this molecular fragment, then we got a partial agonist, a weak partial agonist. However, with another fragment located in the secondary binding packet, we identified an antagonist again. And pretty much the same was observed in the case of another molecule. This dichlorphenylpiperazine moiety mimics dopamine and sits at the orthosteric binding site. It is a partial agonist. However, with this fragment located in the secondary binding packet, we were able to turn a partial agonist to a full agonist. And this one, again, it was a partial agonist. So the pattern is basically the same. You could identify that with this particular SBP binding moiety, we were able to manipulate the functional activity from the secondary side. Oh, what's happened here? Because typically functional activity is modulated from the orthosteric side. This is how exactly dopamine works, right? So we try to understand what's going on here. And the trick is that just because of the different interactions formed within the secondary binding site, these interactions finally result in different orientations of the aromatic rings at the orthosteric binding site. And this is how we can modulate the functional activity, so through the structure of the molecule, actually through the linker, just adjusting a different conformation for the orthosteric binder. Okay, we thought that let's see whether or not it works for another GPCR. Okay, this was 5-HT2A. We found this compound alone as a full agonist, but the two other compounds, when trying to extend towards the secondary binding site, these compounds were totally inactive. Okay, what happens here? This molecule is located in the orthosteric binding site, is a partial agonist, but look, virtually nothing happened here. I mean, in functional terms, because attaching these two secondary binding packet binders, basically we receive pretty much the same partial agonistic activity. So the problem is that these molecules, however, these are basically the same molecules, but the receptors are not the same. And interestingly, this molecule here, that is actually coriprozin, that was found to be in a very strange binding conformation. This is upside down if you compare to the conformation we observed in the case of the dopamine receptor. And the problem is that there is an empty space around the orthosteric binding site of the 5-HT2A receptor, and we can observe an octadecanonic acid that is located within the orthosteric binding site and also influence the upper part of the secondary binding packet that simply prevent the proper orientation of cariprazine in the case of this receptor. So we were not absolutely sure about that, so that's why we did an experimental validation, and we prepared two types of cariprazines. One cariprazine was generated from the original compound, attaching a linker and a large fluorescent dye to this part of the molecule that is typically in the secondary binding pocket. And the other compound, this is fluo-2 cariprazine, we attach the linker to the autosteric binding moiety of cariprazine and using the same dye. And we tested the functional activity of these two compounds against D3 and 5-HT2A receptor, and we found that the molecule that we use labeling the secondary binding packet moiety, it bound basically to the D3 receptor a much higher potency as compared to the 5-HT2A receptor, just because the large dye is now outside from the receptor. And in the case of the other one, when we labeled the autosteric part of the molecule, then we observed better potency in the case of the 5-HT2A receptor as compared to the D3, just because in this case, this is the part of the molecule that is located closer to the extracellular part of the receptor. So at the end of my talk, I would like to summarize what we found. We think that the extending molecules toward the secondary binding site would be a wonderful option to generate GPCR ligands with desired pharmacology. So just because in the case of the class A receptor, the secondary binding packet is transient and metastable, that's why we developed this computational methodology coupled with the experimental screening of our fragments to identify the best combination of the OBP and SBP binders, linking them and developing new compounds against different receptors. So this is also an indication that we can develop these bitopic compounds from molecular fragments, just in the case of LIGO blocks. So we can generate something useful using the LIGO approach for GPCR receptors. We also demonstrated that from the SBP, we were able to influence the potency, the selectivity, functional activity, and also the functional selectivity of the molecules. That would be quite a range of pharmacological interventions we can do in a designed way. At the end of my talk, I would like to acknowledge all of the people who contributed to this work, and thank you very much for your kind attention.